All right, well, thank you, Natalie, for the kind introduction, and also to the ASBMB for this honor. I'm certainly grateful. So as was mentioned, my lab is in general interested in how cells adapt to mitochondrial dysfunction. We got into this years ago when I was a postdoc, but I've continued doing this in my own lab for the last 6 or 7 years. We got into this for, I would say, a pretty naive reason. Mostly that there's a number of human pathologies and conditions, such as aging, that are associated in some way with defects of this organelle that's typically associated with metabolism. So you find a number of cancers with sort of crazy metabolic phenotypes, in this case, an incredible overproliferation of mitochondria. Certainly in Parkinson's and other neurodegenerative diseases, mitochondrial dysfunction probably contributes to the pathology. You see this in aging, and turns out you actually see it in a number of bacterial infections as well. But at the time, and still currently, we don't really have a great understanding of what cells are trying to do, sort of endogenous pathways that are there to sort of monitor mitochondrial function and respond accordingly and try to get back the activity that is faltering. And so in the next 20 minutes or so, I'm going to try to cover a couple of topics that we've done in the last several years, sort of talk about a pathway. So how do cells evaluate this pile of organelles? So these are, these bean-shaped structures that are important for a number of metabolic events, the TCA cycle, oxidative phosphorylation, et cetera. But these things are constantly dividing and moving about the cytosol, and so how does the cell keep track of these things? And if they're not functioning appropriately, talk to the nucleus to turn on genes or transcripts to sort of repair and recover the mitochondrial function. And so this is working out a signal transduction system, and then in the last half of the talk, I'm going to talk about a sort of an unexpected physiological role for such a pathway during the infection by bacteria that specifically target mitochondrial activity to promote their own infection. All right, so over the last 4 or 5 years, we've come to the conclusion that essentially what we're doing is studying a pathway that sort of, how do cells count mitochondria? This is, that pretty much the first sentence of every biochemistry textbook. The section on mitochondria is something to the effect of each cell type, depending on its physiology, has enough mitochondria to keep those cells going. And so typically oxidative cells, such as muscles and neurons, have more mitochondria than cells such as fibroblasts or certainly sperm and red blood cells, which don't have any mitochondria. And there's certainly biogenesis pathways in place to sort of make mitochondria or reproduce them, and this is sort of antagonized by degradation pathways, and then the cell gets to the amount of mitochondria that it needs to do whatever physiological activity that it actually has. And so what I want to talk about today is if the cell determines this number is wrong, are there compensatory pathways in place to get the mitochondrial activity back to what that cell needs to do its job in the body? And so just by way of introduction, I want to give you one slide on mitochondrial biogenesis because it bears on the signal transduction pathway that we've been looking at. So mitochondria are, these organelles that are strung throughout the cytosol here in yellow, and they're comprised of about 1,200 proteins. 99% or so are encoded by nuclear genes, translated on cytosolic ribosomes, and targeted to each one of these compartments, typically by a signal sequence in the protein, typically at the N-terminus here. In this case, it's an amphipathic helix with a number of charged residues. And this sequence gets it to the outer membrane of mitochondria where it interacts with this channel called the Tom channel. The major point here of this slide, though, is that in order for the protein to cross the mitochondrial inner membrane and get to its appropriate location, this event requires just about every metabolic activity that we know that mitochondria do. So to cross the inner membrane requires the TCA cycle, it requires oxidative phosphorylation, it requires ATP, it requires mitochondrial chaperones here on the inside. And so if something is wrong with the metabolic aspects of mitochondria, import efficiency is reduced. It doesn't have to be blocked, but it's simply reduced. And so import is coupled to mitochondrial activity very, very tightly. All right. So here's how do cells monitor mitochondrial function, and if something's wrong, how do they talk to the nucleus to turn on a protective response? And so some years ago, we generated, I should point out we did these experiments initially in C. elegans because at the time genetics were cheap. And the assay is quite nice in C. elegans, as you can perhaps see from here. So we wanted to study transcriptional adaptations, and so we identified a gene, in this case HSP60. It's a mitochondrial chaperone encoded by a nuclear gene, and we put this promoter in front of GFP. In healthy animals, the promoter is on at some low level, but if you perturb mitochondrial activity by just about anything you can imagine, in this case we're knocking down a mitochondrial protease, but if you inhibit a respiratory chain subunit, you get the same effect. this promoter is activated and this worm becomes flaming green. And so we use this assay to screen all the genes in C. elegans looking for components required for signal transduction. So what genes are required for this GFP to turn on? And we screened them all and we found 5 or 6 proteins. The one I'm going to talk about today is this novel transcription factor that we called ATFS1. And so despite high levels of mitochondrial dysfunction, this pathway cannot be activated in the absence of ATFS1. And so ATFS1 is a transcription factor, has a DNA binding domain, a leucine zipper, and a nuclear, like most of them, a nuclear localization sequence. And perhaps not surprising, during mitochondrial stress, ATFS1 accumulates in the nuclei of these 2 cells, where it's involved in transcribing at least this reporter gene. But the question is how is it regulated? You can see in the absence of mitochondrial stress when the worms are healthy, it's not in the nucleus at all. So it's regulated in some manner. How is this? What stimulates ATFS1 to go to the nucleus? And so we started thinking about how to figure this out, and we did a number of biochemical experiments trying to figure out where ATFS1 is located in the absence of stress. Perhaps this will give us a clue as to how cells are monitoring mitochondrial dysfunction or mitochondrial function, as the case may be. So a postdoc in the lab identified at the N-terminus of this protein, what looks like a mitochondrial targeting sequence. You can see a handful of arginines here and enough hydrophobic residues to think it forms an amphipathic helix. And sure enough, the first 100 amino acids of ATFS1 are sufficient to get GFP into mitochondria. So this is cool. It's going to the right organelle. You can see here, it looks like a normal mitochondrial targeting sequence. When it's translated into cytosol, it's of a certain molecular weight, and upon import, the signal sequence is cleaved. So it gets into the mitochondrial matrix. This is topologically complicated, though, right? Being buried in the mitochondrial matrix is a long way from the nucleus, and so what's actually going on? So we made a number of antibodies, most of which failed, for reasons that are complicated, I suppose. But anyway, you can see from this plot here that on wild-type lysates compared to ATFS1 deletion lysates, you never see a band in the wild-type lane that's not in the deletion lane. And so it suggests that our antibodies don't work, or perhaps the protein has a short half-life. And so here on the right is perhaps a poor man's pulse chase, if you are imaginative. The promoter of ATFS1 driving GFP expression. See, this worm is flaming green. The promoter is strong. You take the same promoter and drive expression of an ATFS1 GFP fusion protein, and you can see the animal is basically dark, suggesting that the protein perhaps is turned over relatively quickly. And so one of the nice things about worms is you can quickly knock down a number of genes, and so ATFS1 has a Myto targeting sequence and perhaps a short half-life. Perhaps it's degraded by one of the mitochondrial proteases that are very well characterized. And so we knocked down all of these. And fortunately enough, knockdown of this protease LON, it's a quality control protease inside of the mitochondrial matrix, stabilizes the fusion protein and actually stabilizes the endogenous protein here in the mitochondrial matrix. And so this suggests this sort of odd biology that this protein is expressed at high levels throughout development. The Myto targeting sequence is dominant for, I would say, mysterious reasons, but it's efficiently imported, signal sequence is cleaved, and it's turned over by this protease LON. This seems relatively inefficient, but it's not unlike P53 or HIF1, which are constantly made and turned over by proteasomes. The cool part here is that this is coupled to mitochondrial protein import. All right, so what happens during Myto stress when we know ATFS1's in the nucleus? See here on this particular Western blot, ATFS1 still gets imported. You see some accumulation here in the matrix when you activate mitochondrial stress that activates the pathway. But the stuff that accumulates in the nucleus up here at the top of this gel is of a higher molecular weight than what you see inside of mitochondria. And so this suggests that the signal sequence is still intact, which means the stuff in the nucleus was never ever imported into mitochondria, as the signal sequence was never cleaved. And so this suggests, I think, a pretty simple model. In the absence of stress, the mitochondria are fine. This protein is targeted to mitochondria. It's imported very much efficiently. Signal sequence is cleaved. The protein is turned over. But if something is wrong with mitochondria, import efficiency is reduced. And perhaps because the NLS is allowed to fold in the cytosol, it can now traffic to the nucleus to turn on a response. So to support this sort of idea, we did a number of different experiments. I'll show you a couple here. If you simply take off the Myto targeting sequence of ATFS1 and express the protein, you can see it, basically every single molecule ends up in the nucleus when you remove the Myto targeting sequence. So lack of going to the mitochondria is sufficient to get everything into the nucleus. Probably more interesting, lack of mitochondrial targeting is sufficient to turn on the entire mitochondrial UPR. Mitochondria are fine in this case. In this case, what we did was we took worms that lack endogenous ATVS1, put back in full-length protein, nothing happens as expected. You put back in the protein that lacks the Myto targeting sequence, it all goes to the nucleus and turns on the pathway to flaming green levels. And this, not surprisingly, requires the nuclear localization sequence. And so in sum, we think this sort of data suggests that what cells are doing is monitoring mitochondrial import efficiency. So this protein is constantly made, it's constantly imported, and constantly turned over. But if something is wrong with mitochondria, oxidative phosphorylation damage, chaperone capacity is overwhelmed, high levels of reactive oxygen, import's reduced a bit. This allows ATVS1 to accumulate in the cytosol and the NLS to take the protein to the nucleus to turn on a protective response. And so what we think that the cell is doing is simply monitoring mitochondrial protein import capacity or counting import capacity of this entire pool of organelles. It sends out ATVS1 is something of a probe, perhaps, and if the capacity of this network is good enough to take it all up, everything is fine. But if something is wrong with mitochondria, ATVS1 accumulates in the cytosol, and it comes back to the nucleus in proportion to the amount of stress that's there. And so what genes does it turn on? And we found about 500 or 600 genes that ATVS1 modulates the transcription of, and I'll go through this quite quickly. They fall into a number of categories, typically mitochondrial protective. Via RNA-seq and some microarrays, and more recently a ChIP-seq experiment, ATVS1 binds a 9 base pair promoter element, in the promoter of this case a mitochondrial chaperone gene, induces its expression, presumably to promote the folding and biogenesis of newly made or newly synthesized mitochondrial proteins. So we've got a lot of things involved in mitochondrial proteostasis. Another category, we found many metabolic adaptations, most of which I can't understand at this point in time. But the ones that do make a little bit of sense are the glycolysis genes. So ATVS1 binds the promoter, every glycolysis gene, and activates its transcription. What we think this means is that if oxidative phosphorylation is in decline due to mitochondrial stress, you turn on a pathway that makes ATP, in this case in the cytosol, independent of mitochondria, to keep energetic levels high enough to maintain cellular life and then perhaps recover and rebuild mitochondrial activity. In the last, we found a number of genes that look like a sort of a mitochondrial biogenesis pathway, which makes sense to sort of repair and recover those dysfunctional organelles so the cell can get back to doing what it is supposed to be doing. So in the end, we think at least half of the genes that are turned on by ATVS1 sort of fall into this category of repair and recovery of dysfunctional mitochondria. You also get a lot of genes that are sort of involved in not dying during the process, which I think probably makes sense as well to give the cell time to sort of rebuild this network of dysfunctional organelles. And so what is this pathway good for? This is what my lab primarily does now, and I gotta say, it's not that easy to do, but we've got a number of clues over the years. So consistent with ATVS1 having a very short half-life, animals lacking ATVS1 under normal conditions are basically fine. A little bit of a germline defect, but they grow and lay eggs and propagate just fine. But this is a mutant worm. It's ISP1 mutant is one of the iron sulfur cluster proteins in complex 3, so they have a defective respiratory chain and they're quite sick. But if you knock down ATVS1 here, these guys simply can't develop at all, suggesting that this protein is required, this pathway is required for development during mitochondrial dysfunction. This is definitely not what this pathway evolved to do, as this worm is certainly incapable of living outside of my laboratory, but it is consistent with the idea that ATVS1 promotes development or resistance to mitochondrial dysfunction. So in the last 5 minutes or so, I'm going to shift over to something that was a complete surprise coming out of these microarray experiments. So in addition to a mitochondrial protective pathway, ATVS1 turned on a number of genes involved in detoxifying foreign molecules, so xenobiotic detoxification, as well as a number of innate immune genes, so genes involved in killing off microbes, antimicrobial peptides, as well as secreted lysozymes. This was, I think, it was curious, certainly to us. Why would you have a mitochondrial protective response or a couple of mitoprotective response in an innate immune response by a single transcription factor that responds to decreased mitochondrial import efficiency or mitochondrial stress? If you have an OX-phos perturbation, you can imagine turning on this sort of pathway to keep the organelle functioning to sustain life, but why couple it to an innate immune response? Thought about this for a long time, and this is the general hypothesis that we've come up with. There's a number of ways to detect pathogenic bacteria or foreign microbes in metazoans, including direct recognition of LPS or flagellin or some sort of microbial component. In the case of an environment like the intestine where there's lots of these ingredients, lots of LPS, lots of flagellin, how do you differentiate the toxic from the commensal type bacteria? And so perhaps this is one of what's going on here, is that perhaps if pathogens perturb mitochondrial function, you get this sort of response to keep the organelle working, then the innate immune response to sort of limit the pathogen growth. And this sort of idea was pretty pie in the sky, and then we realized that all the molecules we buy from Sigma to perturb mitochondrial activity, such as antimycin and oligomycin, actually are made by bacteria. So this is made by streptomyces, which are not pathogens, but they are super toxic. And recall from sort of evolutionary biology that mitochondria are derived from bacteria, from prokaryotes. So they have this sort of machinery or this sort of warfare probably works just fine fighting against metazoan species that have mitochondria as well. And so the idea is simply that if there's a pathogen that makes a toxin to perturb mitochondrial activity, the cell probably, it turns on a mitoprotective pathway, but perhaps evolution is just, hedging and assuming this is coming from some sort of pathogen, such as bacteria. And I should point out, a number of viruses and a number of bacteria are known to make mitochondrial toxins, one of which that we studied this, sort of tested this hypothesis with, was looking at Pseudomonas aeruginosa. So this is a human pathogen that grows easily enough to feed it to worms. It typically infects patients that are sort of immunocompromised, such as burn victims or chemotherapy treatments. But the way Pseudomonas kills C. elegans is it accumulates in the intestinal lumen and eventually lyses the intestinal cells. And so as I mentioned before, during Myto stress, these innate immune genes are induced, but if you put worms on a pathogenic strain or a patient isolate of Pseudomonas, you get the same sort of induction of this secreted peptide, the secreted lysozyme, as well as a few lectins that are involved in bacterial recognition. And they all require ATFS1 activation. So as I told you, Pseudomonas accumulates in the C. elegans gut. This is exactly the place where the response is turned on. So this secreted lysozyme here is turned on only in these 20 intestinal cells, and this is blocked when you knock out the transcription factor ATFS1. So along with the innate immune response, the mitochondrial protective response, in this case the mitochondrial chaperone, is also turned on in these same 20 cells, which is blocked when you knock down the ATFS1 protein. All right, so does Pseudomonas perturb mitochondrial function? Actually, let me go back for a second. One of the highlights of Pseudomonas is also that the virulence response has worked out quite well, and one of its virulence factors is cyanide. Cyanide is a complex 4 inhibitor of the respiratory chain, and so perhaps this is a good way to detect these types of pathogens. All right, I'll go through this quite quickly, but it looks like Pseudomonas causes mitochondrial dysfunction consistent with it making cyanide. For example, this is looking at the mitochondrial structure in the intestinal cells. Normally there's sort of a punctate structure throughout the cytosol, but during mitochondrial dysfunction, by knocking down a protease or feeding the worm Pseudomonas, you get this sort of elongated mitochondrial architecture. And then if you put worms on Pseudomonas, in addition to low levels of mitochondrial toxins, they really can't grow at all. On E. coli, they grow just fine despite the low level of toxins, suggesting that something that Pseudomonas does is perturbing mitochondrial activity. I pointed out earlier that the virulence response in Pseudomonas has worked out pretty well. It turns on about 500 genes during infection, and they're all regulated by this two-component system, GAC-A and GAC-S. And if you take away GAC-A from Pseudomonas, they're no longer synthetic toxic with mitochondrial poison. It's just something about the virulence response targets mitochondria. I think most impressive here is that feeding worms that lack GAC-A, Pseudomonas that lack GAC-A, that lack a virulence response, the Myto UPR is no longer activated here, suggesting that the virulence response is what, in this case, this pathway is detecting. Probably more interesting, if you knock out the cyanide synthase operon from Pseudomonas, the worms no longer detect the bacteria in this case. So lacking cyanide synthase, the pathway is no longer turned on, and the worms are no longer sick on this particular bacteria. And so does this matter? Does ATFS1 function have any importance here? As I sort of mentioned, lifespan when you knock down ATFS1 on E. coli, they live pretty much a normal lifespan. But on Pseudomonas, the worms are quite sick, but lacking ATFS1, they die about a day faster, suggesting that this pathway is protective during exposure to this particular pathogen. But this experiment doesn't tell you if it's actually because of a mitoprotective effect or if ATFS1 has bona fide innate immune activity. So we did one experiment, or a couple experiments to look at this, one of which is derived here. We got a hold of a strain that has constitutively active ATFS1 because it has a point mutation in the Myto targeting sequence. So this worm, the pathway is constitutively active in this worm, so you get lots of mitoprotective genes, but also lots of increased in antimicrobial genes as well. And so what we did was we put wild-type worms on Pseudomonas expressing GFP, and within a day or so, they colonize the gut. If you pre-activate the pathway, they're quite resistant to the Pseudomonas. It takes about a day and a half longer for the accumulation to occur, and this requires ATVS1. If you knock it down there, Pseudomonas comes back. And this gives you about a day and a half extra lifespan. And so this suggests that something that ATVS1 is doing is either killing off the bacteria or promoting flux through the intestine. So it seems like it has bona fide innate immune activity. And so this is our current model. One of the reasons this pathway perhaps evolved was to respond to pathogenic or toxic bacteria that make a number of toxins. So I mentioned Pseudomonas, but Helicobacter makes a pore-forming protein that perturbs mitochondria. A number of viruses also attack mitochondria. And so I think this pathway turns on its mitoprotective effect to survive toxin exposure in an innate immune response in case that toxin was coming from a pathogenic or toxic bacteria. And so our idea is this is one way to differentiate between good or toxic and non-toxic bacteria. And so the only experiment we actually have that suggests this is done in Gary Rufkin's lab pretty recently as well, where he got a hold of 600 bacteria from natural isolates from French dirt, basically, and fed each of these guys to this Myto UPR reporter worm that we had generated. And you can see here that something like 18% activate the response. The other 82% don't. Pseudomonas is on this list, as are a couple of other bacteria with cousins, essentially, that are human pathogens. So maybe this is a good way to differentiate toxic and non-toxic bacteria in complicated environments such as the skin or the intestinal lumen. And so let me just thank the people who've done this work. The signal transduction stuff at the beginning was done almost entirely by Amrita Nargan, and the Pseudomonas work was done by Mark Pellegrino, a postdoc in a lab who just took a job elsewhere, and with help from a graduate student, Chris Biarese. And so I'd like to thank the ASBMB and you for your attention.